Hello, my name is Hannah Lynch, and I will be reviewing the Huntington polyglutamine dependent protein aggregation in reconstituted cells journal article. The authors are right below the title, and I'm going to jump right in. The purpose of this journal article was essentially that they implemented polyglutamine, otherwise known as polyQ sequences, into GUV systems. So essentially, they just took previous in vivo research about these polyglutamine sequences, showing that they form aggregation and symptoms of Huntington's disease, and then they implemented them into an in vitro system like the GUV system, so it's not living. The two different types of systems they used were the HeLa cell extract with the human CFPS system, which was essentially this in vitro liposome structure, and it had some cellular components to it in order to help with the translation and transcription process of those sequences. And then the other one they used was even more simple. It had a purified human factor, which was essentially a reconstituted form of the first one. So throughout all of these experiments that they did in the article, it showed that the GUV systems could serve as a simplified basis to study neurological diseases with, just like Huntington's. Now in figure 2, on the top hand corner, we have the EGFPHA sequence, and that has no polyQ repeats, so it is the control group. In the middle, we have an N-terminus of 17 and then 25 Q repeats. And then at the bottom, we have another N-terminus with 96 Q repeats. On the top, with the control, we suspect that there will be no aggregation for the proteins since there are no polyQ repeats. In the middle, it is well below the threshold of Q repeats, and so we also suspect it'll have the same results as the control. And on the bottom, it is well above the threshold for Huntington's with polyQ repeats, and so we suspect that there would be aggregation in those proteins. On the left column, BF stands for bright field, and that is just showing the outside lining of those liposome structures. In the middle, we have GFP, which is the fluorescence, and then on the right, we have merge, which is the superimposed image of both of those. Now, if you look in the middle at GFP, we see that the top two columns have a uniformly distributed fluorescence throughout those structures. It is very dim, and you could individually see the tagged proteins. Whereas at the bottom, with the 96Q repeat, we see very intense fluorescence and small structures within the liposome GUV systems. Comparing those two, we see that the last one actually does have aggregation because those proteins are fluorescing in a really bright way. And on the right-hand side, those are seen within those cell structures of Brightview. Now, these are done with a HeLa extract-derived CFPS system, and so it's a slightly more complicated one that has some cellular components to it. And although it is in vitro, and they were testing in this first experiment in order to see if this aggregation does happen in vitro systems, it was confirmed, but it doesn't necessarily say that aggregation is an intrinsic property of the polyQ repeats. And this is because since there are other cellular components like proteins and whatnot inside these GUV structures that they used the first time, they might have created additional assistance in order to help those polyQ repeats eventually cause aggregation. So it did help the scientists establish a basis for the paper, but it did not confirm that those can happen all the time in an in vitro system without those cellular components. So in the future experiments, they used, as well as the HeLa cell extract GUV system, the other more simple one with just the purified parts. So the final thoughts on this are that they showed that this aggregation can happen in a simple in vitro GUV system, as seen in figure 2, and throughout other experiments within the journal article. And it also helped the field develop a clarified understanding on specific mutations. For instance, having this biological basis being studied in an in vitro system was able to help them fully understand and demystify the components and also the interactions with the polyQ repeats and aggregation in general. Here's the reference, and thank you for listening.